And I am absolutely thrilled to have you here. Today, we are totally rethinking how we look at carbohydrates. I mean, honestly, forget just thinking of them as the bread or pasta on your plate. In this explainer, we're going to explore carbs as these fascinating, microscopic, biological Lego blocks. They are literally the engines of life. They build entire forests, they fuel our brains, and they dictate the very flow of energy on Earth. Let's get into it.
Okay, let's dive right in. Here's our roadmap for today. First, we'll meet the carbohydrates. Then we'll hit the lab for the Ozone sugar test, look at linking sugars together, explore complex polysaccharides, and finally, check out glycolysis and gluconeogenesis. All right, kicking things off with Section 1, meet the carbohydrates, the blueprints of energy. So at their most basic chemical level, carbohydrates are exactly what the name implies, right? Carbon and water. They follow this really simple empirical formula, CH2O taken N times, where N is 3 or more. It's wild to think about, but these molecules are the primary way nature stores fuel and builds structures. It's elegant, it's simple, and it is just incredibly versatile. And what's cool is how these basic carbon-water units scale up. Like, dramatically. We start on the single base level with monosaccharides. Those are your simple sugars like glucose, fructose, and galactose. But, they don't stay single for long. They snap together into two sugar molecules called disaccharides. They scale up into oligosaccharides, and eventually they form these massive complex chains called polysaccharides, stuff like starch and cellulose, which can literally contain thousands of sugar units. Now, here is what's really fascinating. Identical ingredients can actually create entirely different sugars. Take glucose, galactose, and fructose. They all share the exact same chemical formula, C6H12O6. They're all hexoses, meaning they have 6 carbon atoms. But look at how they're arranged. Carbohydrates with an aldehyde group at the end of their chain are called aldoses, like glucose and galactose. But if that ketone group is tucked in the middle, like in fructose, that's a ketose. That tiny, slight rearrangement of atoms completely changes how these molecules behave and react. It's like having the exact same Lego bricks, but building two totally different spaceships. Moving right along to Section 2, the Ozone sugar test, basically our detective lab. So you might be wondering, how do we actually tell these invisible, identically formulated sugars apart in a real lab? Well, our source material introduces this wonderful analogy of a Madame Moira playing with magical crystal balls that change color and shape based on the ingredients she uses. And honestly, in the chemistry lab, this magic is very real. We use predictable, observable reactions to unmask these invisible sugars. This specific kind of magic is called the Ozone test. When reducing sugars are boiled with a reagent called phenylhydrazine in an acidic buffer, a really fascinating reaction goes down. The phenylhydrazine specifically targets and reacts with the free carbonyl groups of the carbohydrate. Give it some time and high heat, and this interaction creates a derivative compound known as an ozone. And the absolute best part? These ozones form vividly colored, physically distinct crystals. Just look at this data. It's essentially a fingerprint database for sugars. We can identify the exact sugar just based on the shape of the crystal and the time it takes to appear. For instance, fructose produces these needle-shaped crystals in just 2 minutes, super fast. But if you're dealing with lactose, you'll be waiting a good 30 to 45 minutes to see crystals that actually look like little cotton balls or powder puffs. And galactose forms these wild, thorny balls after 20 minutes. It's just a brilliant, highly visual way for scientists to tell different reducing sugars apart. Which brings us to Section 3, linking sugars together, or building the bricks. So, the secret to linking these sugars together lies in a very specific chemical reaction. When the aldehyde group of one molecule reacts with the alcohol group of another, they form what's known as a hemiacetyl bond. In the carbohydrate world, this connection between sugar units is called a glycosidic bond. Think of it as the biological mortar holding our microscopic sugar bricks together. And here is the result of those linkages, the disaccharides. Look at how versatile D-glucose is as a building block. If you link a glucose to another glucose, boom, you get maltose. Link a glucose to a fructose and you get sucrose, which is your standard table sugar. Link galactose to a glucose and you've got lactose, the sugar found in milk. By simply mixing and matching these basic monosaccharides through those glycosidic bonds, nature creates all these diverse, crystalline, water-soluble sugars we use every single day. Next up, Section 4, Complex Polysaccharides, Nature's Architecture.
Let's take a look at plant starch. This is the carbohydrate reserve for plants, and frankly, a massive energy source for us. Now, starch is a homopolymer, meaning it's made entirely of repeating glucose units, but it actually comes in two different structural forms. First, we have amylose, making up about 20% of starch. It's a straight linear chain, and it dissolves easily in water. Then we have amylopectin, making up the remaining 80%. Unlike amylose, amylopectin is highly branched, and it is completely water-insoluble.
You can picture that linear amylose perfectly held together by straight-shot alpha-1,4 glycosidic bonds. But check out the amylopectin. Every so often, it throws in an alpha-1,6 glycosidic bond, which creates a distinct angled branch. Because of that, a single amylopectin molecule can contain thousands of glucose units branching out, literally, like a microscopic tree. It's just an incredible piece of biological engineering.
Now, if you change that bond just slightly, you get something entirely different. Cellulose. Like amylose, cellulose is a linear, unbranched polymer of glucose. But instead of alpha linkages, cellulose uses beta-1,4 linkages. This creates a really rigid, tough, fibrous structure instead of a squishy energy storage molecule. It forms the cell walls of plants, is quite literally one of the most abundant organic compounds in the entire biosphere. So just changing the angle of the chemical bond turns food into wood. How crazy is that? All right, final chapter, Section 5, Glycolysis and Gluconeogenesis, the engine room. Enter glycolysis, sometimes called the Embden-Meyerhof pathway. This pathway is the central hub of glucose metabolism and the biggest flux of carbon in almost every single cell. It is a precise, beautiful 10-step enzymatic process perfectly designed to degrade one 6-carbon molecule of glucose into two 3-carbon molecules of pyruvate. Now, please don't let that 10-step process overwhelm you. The really crucial takeaway here is that glycolysis is divided into two elegant phases. First up is the preparatory phase. Here, the cell actually spends energy, investing 2 molecules of ATP to phosphorylate and prime the glucose. But trust me, this investment is totally worth it. In the payoff phase, that primed molecule is split and oxidized, paying off the debt and yielding a biological profit. The net result from just one glucose molecule? 2 pyruvates, 2 NADHs, and a net gain of 2 ATPs. Basically, you gotta spend money to make money, biologically speaking. But what happens when our immediate free glucose runs out? Say you're halfway through a really long run. Well, our body taps into its emergency reserves, glycogen, which is structured a whole lot like that plant amylopectin we talked about. An enzyme called glycogen phosphorylase acts repetitively on the ends of these branches, snipping off glucose 1-phosphate molecules one by one. And once it gets a bit too close to a branch point, a debranching enzyme steps right in to clear the path. That allows the breakdown to continue, keeping our cellular engines running smoothly. But honestly, our bodies are even smarter than that. If we completely exhaust our carbohydrate reserves, like during extreme vigorous exercise, the body can literally run the machinery in reverse, sort of. This is called gluconeogenesis, the generation of new glucose. It salvages non-carbohydrate precursors, like the lactate that's building up in your anaerobic muscles, and painstakingly converts it back into glucose to keep the brain and body alive. Glycolysis burns the fuel, right? But gluconeogenesis essentially rebuilds the fuel from the exhaust. It is mind-blowing. Which brings us to a really fascinating conclusion. Carbohydrates are an incredible study in duality. On one hand, they're the structural framework of the natural world, the sturdy cellulose holding up towering trees. On the other hand, they are highly volatile fuel, just waiting to be aggressively broken down in a 10-step metabolic engine to power our very thoughts and movements. It really leaves you wondering, if biology can build complex life from simple sugars and aggressively burn those same sugars to sustain it, what other future biochemical miracles might we be able to unlock as we continue to master these basic blueprints? Thank you so much for exploring this with me today. Keep questioning, keep learning, and I'll catch you in the next explainer.